Okay. Okay. I think we almost ready for the class. Okay, so this is for this class is just for the brief introduction of the Drosophila genetics. So this PDF file is our current textbook. It's a rough guide to Drosophila mating scheme. It's not only for the Drosophila mating schemes, but it also contains the kind of the basic concept of the Drosophila genetics. Okay, so it has the contents, a lot of contents in there. So I will starting from the most basic step. Okay. Okay. So boxes and figures. Why work with the fruit fly? Why work with the Drosophila melanogaster? So there's a history of the Drosophila research. So we need a model system in the biology. So in theory, so why we study biology is we want to know the human being and our kind of the mechanism to the how our living system is working, especially human. So, and there's a two clear aims. The first aim is making the drug for human to cure human disease. And second aim is just curiosity for the living thing in the life, in our earth or in our life area. So the first aim is our department is focused on the first aim, literally. So we just call the 분자 생명과학 or molecular biology. Is usually focused on the first step is understanding the human being. Second step is pinpointing or point out which step or which molecule and which kind of the gene is important for the disease. And third step is making some drug for curing and solve the issue on the gene, protein, and those things. That is a kind of the one aim of the our life science. Second aim is it is quite romantic. So we need to know the how many animals live in our country. So that somebody need to check up how many animals, for example, how many species of the frog live in South Korea. Our government need to do that. And how many trees, how many species of trees, and how many species of the birds, and their translocation, and how many species that survive. Somebody need to do that. There's eco biology, eco life science, also in our department, one part of our department. We can call there's an eco department, ecobiology. So we have one of famous Korean scientists for the ecobiology, Choi Jae-chun, kind of the very famous celeb in the South Korea. So he is focused on those things. So, but our department is focused on the making the drug for curing human disease. And Drosophila is kind of the model system for because it have a similar mechanism, basic mechanism of the life, a living system. So, for example, when you think about for example, in human, there's two complicated system. We cannot pinpoint which gene is important in the curing or solving the system. But in Drosophila, it have a quite simple system. So and simple set up the gene, but almost all those genes are kind of the similar with the human. It's a very surprising point. So there's no totally different thing between human and Drosophila. And middle of the Drosophila and human, there's a mouse is under the model system. So mouse is different from human, but much similar with the human than the Drosophila. So we plan to open up the mouse research in several years. But right now, we focus on Drosophila because it's much more simpler and easy to find out which one is important. So and Yeah, there's history, Nobel Prize using the Drosophila. So Morgan, Thomas Morgan, he know the role of chromosome, and the name, the concept of loci. So the position of the chromosome in the specific position, the specific function is in that loci. So when you just think about which some genetic disease is because of the some mutation on specific area of the chromosome. So that is found by Thomas Morgan. And another famous guy is Richard Axel, kind of the older receptor. So Richard Axel's, I met Richard Axel when I lived in New York City as a postdoc. So in Colombia, so it is another very important finding. So because the odorant receptors and mechanism how to sense the chemical contents in the air is similar with human and Drosophila. So they're all their receptor system and mechanism how to detect a lot of combination of the chemical using the limited number of gene is also homolog between the human and Drosophila. And okay, so Hoffman innate immunity. So innate immunity also very important point because Drosophila also have immune system, and they don't have adaptive immune. They only have innate immune system. And for innate immune research, it is very important and easy model system because a very aligned system, but quite simpler. So we are also in that project too, and it is not updated 2017. So kind of the Michael Rashbash, Thomas Young is finding of the basic principle of the circadian rhythm. So circadian rhythm gene, it's period, timeless. So that kind of the gene control the circadian rhythm. So without that, we cannot sustain our rhythmic life. So we awake in the morning time and we sustain the awakeness during the daytime. And we need to go to sleep and we feel sleepy during around the nighttime, and we can have a kind of the rhythmic sleep time. So Teresa's research point is about the sleep Drosophila, and much more detailed research point is the function of the zinc ion in the fly's sleep behavior. So maintaining sleep behavior. So that is Teresa's main focus on her research, and almost done. The story is almost done. And our lab is focused on the two behaviors. First one is feeding. Second one is sleep behavior. So because of reason our lab name is Behavior Neurophysiology Lab. So basically we care the fly's behavior, and we need to know the neural circuit that evoke the behavior. And that neural circuit is researched using the kind of the neurophysiological tool. So for example, calcium imaging using the Galaxy or GCAM signaling checkup. So that is the neurophysiology. So our lab stuff is connected by two different but very related experiments. First one is check up the behavior changes in flies and find out the mechanism why the behavior has been changed. So finding out the neural circuit changes in fly brain. So using the kind of the neurogenetic or neurophysiological tool. That is about the fly research. And next, Nobel Prize will for the Drosophila. I think it is about the kind of the endocrinological regulation because that is my major. So my major couldn't get Nobel Prize yet in Drosophila. I think I did something and we need to do more in the field, for the field. And okay, let's go to the fly's basic life cycle. Okay, so fruit fly is a very small animal, and it like the fruit because the name is fruit fly. and it sucking the nutrient in the fruit and the yeast, and the kind of a little bit fermented thing. So in Korean name is chopari. Cho is a kind of the sour vinegar. So why is they like vinegar? Is vinegar indicates that there's a little bit the fruit fermentation, and they can be a vinegar. And according to the Greg Sue's lab, Greg Sue was my former PI. According to his Nature paper, fly like acetate, not low pH. That indicates that a fly to get the acetate for their survival, they like vinegar. Anyway, vinegar like fly. So there's a chopari in Korean term because they have they like that thing. In English, the fruit fly because they like fruit as a food source. And there's a life cycle. The female fly looks like this, male fly looks like this. Definitely, female fly is bigger than male flies because they need to laying egg, so they need to store more energy inside their body. So in the feeding behavior, we are using mayflies in the glucose feeding because it's simpler. And we using the female fly for the yeast feeding or sugar versus yeast choice behavior because female fly, especially mated female fly, care the protein. So we care the protein contents in the food because we need to eat the protein for survival. But fly, so there's a two different season in their life. First season is the larval area, larval time. In Korean term is 애벌레. Another more strict term is 부더기. It's a kind of the larva of the fruit fly. So larva season, they need to focus on the grow up. So insect and fruit fly is a very well-designed living organism because they need to separate their life cycle and they need to totally different shape and totally different neural circuit to focus on the aim of the specific season of their life. Growing up season, they don't have sexual organ. They don't develop the sexual organ at all, and they just focus on the eating. At the time, they don't have functional eye because they don't need to see something. Just smell it, chew it, eat it. That is what they need to do then. So larvae, they focus on eating food and grow up. And I suppose they better focusing on the yeast food and protein food. However, in the adult season, they like sugar more because they need to. The number one aim for the late life is a reproduction. So adult. In adult, they have a wing because they need to get more mobility to find out their partner. And so number one mission is the mating, and second mission is survival. So there are some argue with I'm not major in the sexual behavior, but my major in the feeding behavior. I just thought that survival is first and mating is second. But under my colleague, my friend, Uje Kim is a mating behavior expert. He says that mating is first and survival is the second. It's kind of I don't know, because I have kind of the pilot experiment. I start to fly just almost dead. So flies just before the dead point, and I give fly the two choice. First choice is food. Second choice is mating. My pilot experiment result is they choose to eat sugar, especially sugar. They love sugar because sugar is very useful and easy to digest and easy to use and easy to store. So they like it and very rare in the real life. So they love it, like it, more than love it. So according to my experiment, it's like that. And according to Uje's experiment is, when he starved the male fly, he only cared the male flies. He don't care the female fly at all because he only cared the male flies' sexual behavior. When he starved or fed male flies, their sexual behavior is just same. So fed male flies and starved female fly just near to die, their sexual behavior is just same, not altered at all in their kind of energy, internal energy situation. So it was surprising, but in my point of view, survival is first, and then mating. Yeah, that's anyway. So there's a purpose of the adult season, so it's different. So there's a two stage. In more strictly saying, three stages: egg and larva. So when after mating, female fly laying egg, and egg can be after hatching the egg, there's a larva is coming out, and first insta, second insta, third insta larva. So there's a larva. So you already know the insect. Insect has their before and after metamorphosis, and metamorphosis is coming on in the pupa season. So pupa. So pupa, they don't eat, they don't move, they hide in the very cuticle, and they just transform their every body physiological status. They reform their brain, reform their neural circuit, and deform their organ. So there's conducted in the pupal season, and after the pupation, there's an eclosion. And after eclosion, they have adult, female, and male flies. And after the adulthood, they don't have chewing mouths. They have as a straw, one straw in their mouth. They only can sucking the kind of the fluid food. And in the larval season, they have a chewing mouth, so they can chew the thing. So life cycle. So in the 25 degrees of Celsius, egg to adult is egg to eclosion is 10 days. 10 days. So that is our life cycle. So 10 days. 10 days is a very long time. At the same time, it's very short time. Because when you think about the C. elegans, it's a very warm, small worm, nematode. Their life cycle is 3 days. Very fast. And 10 days is not that short, but not that long. And when you think about the mouse, so kind of the small mouse, it is around 4 months. 4 months. So 4 months and 8 months is 4 months is their young season, and after 8 months they can mate. 8 months, 4 months to 8 months. So when you studying your graduate student, after 8 months, you can do the first mating when you grow the mouse in the first place. So it is the fruit fly is quite, and it's individual brain it have basically individual brain system, and they have a brain and spinal cord too. It's not totally like humans brain and spinal cord, but there's a kind of the functional homology. So independent brain and spinal cord, and they have their neuronal system and muscle system, immune system, everything, and not perfectly like human, but a very similarity with human is very highly similarity in the point of the gene. So for example, a lot of the neurodevelopmental genes are shared with the fly and human. So DSCAM or their famous neurodevelopment genes are aligned with the fly and human. So 10 days. Another important point is we can control the temperature and we can control the developmental time. The lower temperature, it takes more time. So for example, when you lower the temperature at 25 degrees Celsius to the 18, it takes this 10 days period can elongate it around around 1 month. 1 month. So, and when you increase to 29 degrees Celsius, it can shorten as short as 6 to 7 days. So, but we do not increase the temperature more than 25 because we're worried about the heat shock damage. But lower temperature, it slows down, it grows. So when we have some kind of the fly lines and we do not using right now, so we keep it in the lower temperature, so it save our money and effort because we transfer the fly. So and do you know what is transfer the fly? So because a transfer means that when you keep the fly in the food bio, we need to transfer the other flies to old bio to new bio because we need to keep them new food sources. So yeah, this life cycle: larva, pupa, adult, and 10 days. And first, second, third is the larva. And okay. And then I need to also teach about the concept of genetic research for then reverse genetic research. So you can read it, but I just briefly let you know in the paper and in the scientific scene, there's only roughly two genetic manipulation. First one is loss of function, and second thing is gain of function. So we know the gene is in the chromosome and in the DNA. So chromosome, the DNA is inside of the chromosome. Chromosome is DNA with the protein and kind of the core protein like histone or something. And in the DNA, there's a gene. So that kind of the segment of the DNA fragment that express containing some information for making some protein. Some kind of the protein can have a function as an enzyme or some kind of the keratin for make a structure or integrin or something like that. So when we have two manipulations, first, get rid of that gene in the animal's body or inhibit or reduce the gene expression, there's a loss of function. And the other opposite way is a gain of function. So increase the expression and introduce more non-existing gene expression, that is a gain of function. Loss of function, gain of function. There's a two main genetic manipulation. So it's quite simple because we have a gene and we can inhibit or it can activate it. But and check what's difference. So get rid of that gene, inhibit the expression of gene, and check what changes in the morphology, shape, behavior. Behavior is our topic. Behavior, characteristic, and every other aspect. So after to get rid of the function, it's loss of function. And gain of function is not existing in that area, but ectopic expression. So we forced to express some gene in more highly amount, and then check what gonna be difference between and after the activation. So many scientists and geneticists says that loss of function is much more natural and gain of function is very unnatural situation because loss of function is usually happen when we got a mutation. Usually when we get mutation some gene, the function of that gene is gone and we have a mutated gene and that make many issue. For example, some people cannot digest some specific amino acid form, ketonuria. So that human cannot digest the ketone body, and they should avoid eating some kind of some stuff in the food. Without that, in their young season, they can be the mental retarded without avoiding that food source because of loss of function, because of the loss of function. Gain of function is not common in the real life, but we love it because the phenotype is very strong. So we easy to write paper using gain of function, but loss of function is much more natural because we can watch a lot of mutants in the real life. There's two things in the genetics. And mating scheme, okay. Fly stock, okay. So in the fly, we have a mutant line. So there's a genome. We have fly genome. Fly have 4 chromosomes. I'll let you a little bit later. And a lot of mutations are made by the former scientists. So using several various ways, so kind of EMS, mutagenesis, we treat some kind of the very harmful chemical that evoke the mutation, and we finding out some mutant happen in the specific gene, and check that gene expression function is just gone, and check some phenotype is happened in the body. That is the mutant line, and mutant line is saved in the stock center. And we also have our own fly stock. So kind of for example, Telsa have a period mutant, which is a circadian rhythm mutant, and also timeless mutant and PDF mutant that is about the circadian rhythm mutant, and Fumin or insomnia, there is a sleep mutant. So because of the mutation of some gene, their sleep length is gonna be shortened than the wild type. So there's a mutation line. And I also have a Piazzo knockoff fly. Piazzo is a kind of mechanosensor in the body. So they eat more because their stomach cannot sense their stomach is enlarged a lot. So there's a mutant line. Okay. First one is mutant line. One gene, there's out of order of the gene. And the second line of the we have line is a transgenic flies. Transgenic fly is introduce some gene part, some kind of a gene fragment inside their genome. So we can usually call this a P element. So P element is jumping gene, basically made by found by the Barbara McClintock in 1928 or something. She got a Nobel Prize because she lived longer. But it's very important because it jumped into the genome and introduced some new function. So transgenic fly. So we introduce new function in the fly using that jumping gene. So it is a little opposite from the loss of function. So just mutated and loss of function. And transgenic fly is introduce new DNA fragment from the outside and inside to their genome. And inside their genome, instead the gene fragment, they have some functional gene is expressed in that area. So they have a new function, additional function. There's a kind of the transgenic. So for example, when we use the wide-eyed mutant background, wide-eyed fly, so it's a mutant. So there's a mutation on the eye color making gene on the X chromosome. But when we insert the P element, and in that P element, we usually contain the W plus gene, white gene, that make a red pigment for the eye. So when P element inserted in the fly genome, the fly have a red-eyed phenotype. The wild type the background of the fly before the P element insertion, they basically have a white-eyed fly, but after the introduction of the new gene fragment, they have a red-eyed phenotype because their red eye making function is introduced from the P element. There is a transgenic lines. And mutant and transgenic, there's two main stock fly line we have usually. There's two main line we need to understand. And handling flies for laboratory. So that is what we need to focus today. Okay, this is vial. So this is a chamber, this is vial. We have 3 chambers in the lab. In our lab hallway, there are 3 big chambers in there. If I have more space, I need to purchase up to 6 to 7 chambers because I have enough money for that. But I don't have enough space for put the chambers. So we have 3 big chambers right now. But you can keep the fly. And the box, you can see the box. We call this box as a tray. One tray. One tray contains 100 vials. This is a vial. It's made by the plastic, and the lower side there's a container food, fly food. So we have a recipe for fly food. We can purchase from the Hansortech, is a very specific company that's selling the fly food. Also, we can make it on our own in the lab in the room 406. There's a fly food making set. Why the fly food making setup is in my room? Because they make smell. And we need to shut my room door, and open all window, and we make the fly food. Because everybody hate that smell. So that is very interlocked in my room. And that would be done by Jasmine. Jasmine will arrive this around today lunchtime, and she is the kind of lab technician. So she gonna making the fly food for us, and also Teresa need another specific type of food, more tiny glass tube, because for sleep measurement, we need to put the fly in the glass tube, and that glass tube contains a food, 5% sugar, 1% alcohol. So that is also made by Jasmine. Okay, so this is the fly food, and this is a tape, and we write down something in there. So this is a kind of the genotype, or the name of the fly line. And we have a cotton flow. So this is the kind of the condensed cotton flow, but we use the condensed cotton flow, so there's no plopping, no the fragment of the cotton flow is floating away, and it go into your inhale, and you do not harm your lung. So we use the condensed cotton flow, and this is a fly stereo microscope for fly watching. So, and this is a CO2 pad. The CO2 is in, you can open the CO2, and CO2 is popping up in here slowly, sound like a sound. And CO2 anesthetize, make don't fly, move, don't move. Bachi, anesthetize. Okay. Uh, and with CO2 gas, fly cannot move. So, and we can watch the fly morphology, male or female, or their balancers and markers. So what we will learn in the next class, that's very important concept in Drosophila genetic understanding, balancers and markers. Basically, markers are mutant phenotype. For example, if you have some mutation in specific area in the second chromosome, then make a wing, they have a curly wing. There's a marker. So we need to marking up specific area of the chromosome using that kind of the specialized mutation. That is, we can call marker that showing the phenotype. Using the phenotype, we can tell what's going on in their chromosome. So there's a marker, so we can check the markers in the fly body. So this is a wild type female fly, red-eyed, straight wing, and we can also catch the kind of the brittle, this hair, the number, and shape, and organization of the hair, eye color, eye shape, wing shape, and body color. That is a kind of we can check the markers in changing the morphology of those specific part. And this is what with CO2 of Drosophila fly looks like this in your night eye. And in using your microscope, you can see like this. So we have a bunch of the microscope in the fly room or in our lab area. And first step in the lab we need to do is learn how to transfer fly. So transfer indicates that there's other flies in here. Move the only other fly to the new fresh bio. That is a transfer. Maybe I already know what it is. And transfer the flies. And the second thing we focus on in the early phase in the lab is to learn how to dissect the brain. So the head, this is a head containing the brain inside the head. And we learn how to brain. We learn how to popping out the brain inside of the head. So there's a brain dissection. So using the forceps, very sharp, kind of the chopstick like knife, we can open the brain cuticle and popping out the brain inside of the head. So that looks complicated and looks very hard, seems very hard, but almost all the people can learn within 2 weeks with my curriculum. But the important point is how to learn that. So first, when you think about you are hired in the sushi house, sushi house, you need to learn how to sharpen your knife. That is the first step. Sharpen your knife first, and you need to learn how to open the brain, how to pop the brain inside of the head. So that is the protocol. Then we have a very well-designed and very detailed protocol we have. So we can learn in short time. Okay. Okay, this is the first step. So, and first step we need to do is segregation, male and female. So I'm gonna stop in here today because male and female differences checking. So I think this is good for today. So this is a male female fly and this is a male flies. So there's a two clearly different point. One point is in their end of their abdomen, in the belly, sexual organ is in the tip of the abdomen. In the male flies, there's a penis in the end of the tip of the abdomen. So it's circle shaped, round circle shaped organ is end of the tip of the abdomen. There is a penis. And in the male female flies, there's no penis. There's looks white in the top tip of the abdomen. That is the most accurate and most easiest way to could segregate male and female. And in the just briefly checking, they're checking abdomen. Black color, much more black color in the male flies, and bright and bigger and more segment in the female fly. And there's an additional one in the tip of the first two leg. There is a sex comb. It's the comb-like structure because this male fly use this organ to grab the female during the mating. So today's what you need to do is segregate male and female, and you need to learn how to using this stereo microscope and how to using this stereo microscope. If you need to first get focus on the microscope and changing the magnitude. So in large view or much more far away view, and train to learn how to do focus and watch your eye using the microscope. and how to use this CO2 pad, and then see the Drosophila and check whether there's a male or female. And today what you need to do is segregation male and female. Maybe I already know how to do that, but just repeat and practice once again. And for the 주경, it is maybe for the first time, but it's not too much. So watch it and tells their end of the abdomen they have this one or this one, and you can tell this one is male, and you can tell this one is female. So that is what we need to do today, and I don't make you force too much thing for today. And we need much more to learn, but we're gonna start today. And after the lunch, around 1 p.m., I arranged the available student, to let you know how to use the microscope and use the CO2 gas and segregate the male and female. So you started from the white type fly first, maybe white 1118, our white-eyed white type fly, and male female segregation, and try and let you train your eye and train your hands for the how to operating the microscope. And the second thing you need to do is read this stuff. So if your English is not good, use a translator. It's okay. Yeah, this nowadays is a good day, so use a translator. But just read it around this page, around just this page, around page 8, around page 9, before this performing cruises. It can be a little bit hard because it explain too much thing in the first place. But anyway, just try. Okay. And if you have a class, you can go to the class. And that's it for today. Okay. 그러면은 여기까지